Hello, everyone. Welcome back to my video tutorials for single-cell RNA sequencing data analysis using the Giotto package. So in our data analysis, sometimes we want to focus on a particular cell clusters and recast the cell clusters to identify the subcell populations. To do that, we need to subset a particular cell cluster, then recast the cells. So today, in my video tutorial, I'm going to show you how to subset a cell cluster from a Giotto object and recast the cells. So we are going to use the NML Giotto project we analyzed in video 1 and video 2. You can see in the data window, we have the NML Giotto object here. Let's plot the cell clusters and have a look at the cell clusters again. You can see we named the cell clusters already. We have the epithelial cells, fibroblast, immune cells, parasite, and smooth muscle cells. So for today's demonstration, I'm going to use the fibroblast cluster. Because in fibrotic lung, fibroblast is the major cell type that produces extracellular matrix. So although this is a data set from healthy human lung, I'm going to use the fibroblast cluster and show you how to subset the fibroblast cluster and recast it in order to identify the subcell population of fibroblast in the human lung. So we can have a look at the metadata first. If we run, you can see in the metadata we have the clusters name column to name all the cell types. And also we have a cluster column named as laden. You can see the cluster numbers for each cell cluster. So if we want to subset the cell cluster by the cluster number, you can use the laden column. We are going to subset the cell cluster by the cluster name fibroblast. So we need to use the cluster names column. So you can see we can use the subset Giotto function to subset the cell clusters that we want to do further analysis. You can see here we can set the cell ID argument as fibroblast, and the cluster column will be cluster names. So we can subset the fibroblast now. Let's run. You can see we created a fibroblast Giotto object. We can run the dim plot function again to double check if we actually just kept all the fibroblast. We can run. You can see now I only have the cell types fibroblast in the new object. Because we already performed the filtering and the data normalization, to recast the fibroblast, we need to calculate the highly variable features again just for fibroblast. In the new object, let's calculate the highly variable features first. Okay, you can see we got the highly variable features for the fibroblast object. You can see again, Giotto package divided the cells into 20 groups and identified the highly variable features for each group. Gene can be used as highly variable features, will be labeled in yellow color, and also S in the metadata. So we got the highly variable features for the fibroblast object. Now we can run PCA for the fibroblast object. Let's run. You can see down here, HVF was found in the feature metadata information. Then Giotto package used the highly variable features to perform principal component analysis. So after PCA, we can use the scRNA plot to identify the best PC number for UMAP analysis. We can run. Let's zoom in. You can see from this table, once the PC reaches to 6, then the percentage of variance explained per PC became flat. So we can use PC6 for UMAP analysis. We can set the dimensions to use the PC1 to P6 and perform UMAP analysis. Let's run UMAP now. So after UMAP, we can create a nearest network. So by default, we are going to create a shared nearest network for the fibroblast object. For the dimension reduction to use will be PCA. Reduction name we can name as PCA. We will use the same dimension PC1 to PC6 for the nearest network analysis. So we can run this code now. So after creating the nearest network, we can use the needing method to identify the cell clusters in the fibroblast object. We can set the resolution as 0.1 for the demonstration. You can use different resolution. The purpose is to identify different subpopulations for your cell type. So let's run leading cluster method to identify the subpopulation for fibroblast in human lung. So after the cell clustering, we can use plot UMAP to look at the cell clusters. Let's run. You can see now we have 9 cell clusters with resolution 0.1 for the fibroblast in healthy human lung. So we have different fibroblast subpopulation now. Then we can perform differential expression analysis to identify the marked genes for each subpopulation and annotate the fibroblast subpopulations. So I'm not going to show you the differential expression analysis and the subpopulation annotation. I showed you the method in my video 2. You can watch my video 2 if you want to perform differential expression analysis and annotate your cell types. Here in this plot, we didn't show the network. If you want to show the network, you can set the show network argument as true to generate a UMAP plot and show the network. This plot takes time to generate, especially when you have lots of cells in your object. So let's run for the demonstration. Okay, you can see we generated the UMAP plot, and you can see the networks between different cell clusters now. So that's it for today's video tutorial. I showed you how to subset a cell cluster from an analyzed Giotto object and reclust the cells. I hope this video can help your data analysis to identify sub-cell populations for your study. So please subscribe to my channel if you haven't to do so, and share my videos with your friends. Thank you.